All right, go ahead and kick some stuff off. Hello, everyone. We're going to get started. Thank you all for joining us for the October 2023 monthly seminar on physical genomics. My name is James Peterson, and I'm the Senior Director of Operations for the Center for Physical Genomics and Engineering here at Northwestern University. For those of you who are new to this seminar, the CPGE is a transdisciplinary research center that studies the structure and function of the genome through the convergence of super-resolution imaging and nanosensing technologies, modeling, computational genomics, biology, and artificial intelligence, with a dual mission to develop new strategies for the treatment of disease and the reversible engineering of living systems, and to train the next generation of multidisciplinary scientists and bioengineers. You can learn more about our research and training programs at the physicalgenomics.northwestern.edu or follow us on YouTube, Twitter/X, or Instagram. We are very excited to welcome as our speaker today, Dr. Andrew Gorkin. Dr. Gorkin is an assistant professor of biology at Emory University, where his lab studies the cell's epigenetic machinery, seeking to understand how this machinery functions during development and how its malfunction can contribute to disease. Dr. Gorkin received his PhD in human genetics from the Johns Hopkins University School of Medicine and completed a postdoctoral fellowship in epigenomics at the University of California, San Diego. Dr. Gorkin, thank you so much for sharing your work with us today. For our audience, there will be time for questions at the conclusion of the talk, and you can just type them, or you can just type them into the Zoom chat at any time, and we'll address them during the Q&A after the talk. At this point, Dr. Gorkin, if you'd like to share your screen, we can begin your presentation. And thank you once again.
Thank you very much. It's an honor to be invited here. Let me go ahead and share my screen. Let's see, Tommy, can you see the title slide here?
Yep, I see it perfect.
Great. Well, thank you. So I'm excited to be here today. I want to tell you about some new directions in my lab, trying to understand chromatin remodeler function in development. And so a lot of the work I'm going to show today is unpublished. It's also still kind of a work in progress. So I'll be looking forward to the Q&A session at the end. I think hopefully there'll be some interesting points of discussion. So outline for today's talk, I'm going to start with some background just about chromatin remodeling and its role, at least what we know of its role in human development and disease broadly. I'll spend the bulk of the time in the middle part here talking about this experimental pipeline that we're building to better understand chromatin remodeler function in vivo in mouse development using mouse as a model system. And then towards the end, I'll tell you about some of our early insights coming out of this pipeline and share with you some very recent data. So first, background. The first point of background that I want to convey is that chromatin remodelers are a class of proteins that my lab is particularly focused on, and I'm going to talk about today, that alter the packaging of DNA around histones. So I'm sure this group is well aware, DNA is wrapped around these histone proteins. This is an octamer of histones to create this nucleosome particle that's about 150 base pairs of DNA wrapped around these 8 histone monomers. And chromatin remodelers are a class of enzymes and enzyme complexes that can move these nucleosomes in different ways. They can evict nucleosomes, they can shift them to the side, they can exchange certain histones for others inside of these nucleosomes. So it's a sort of broad class of proteins and protein complexes. This kind of chromatin remodeling is important in a whole host of different processes. You can imagine that almost any time you need to do anything to DNA inside of a eukaryotic nucleus, you need to remodel chromatin in some way. So if you need to repair DNA, if you need to replicate DNA, all of these processes involve chromatin remodeling in some way. The process that I'm going to focus on most today as a developmental kind of epigenomicist and gene regulation person, at least that's where my main interests lie, is the role of chromatin remodeling in transcriptional regulation. This is really a key step in the process of activating non-coding regulatory sequences like enhancers and promoters. So the kind of canonical model of how we think this works is that the nucleosomes are kind of shifted aside to make the DNA more accessible to the binding of transcription factors and other regulatory factors that can then activate gene transcription. And this happens very proximate to the transcriptional start site at promoters, and also happens distal to the transcriptional start site at enhancers and other types of regulatory elements. So these chromatin remodelers are often function in large complexes, though not always. In total, we know about 100 human genes that encode subunits of these chromatin remodelers. In many cases, these subunits are obligate subunits of these chromatin remodelers. In other words, we don't really find them as monomers. We don't know of functions for them outside of the complex. That's not true of all of the subunits, but it's true of many of them. So those are just some kind of general themes in this area of chromatin remodelers. And to give you a kind of concrete example, I'm going to share with you one of our favorite chromatin remodelers, which goes these days by the name of BAF, also known as switch SNF. This is itself a family of related chromatin remodelers. And this one we think is particularly important in creating this property of accessible chromatin at regulatory sequences like enhancers. And this is a property that we map with a TAC-seq. One of the key pieces of evidence for this came out a few years ago when some groups used a number of means, but in particular a ATPase inhibitor of the BAF complex. BAF uses ATP to shift nucleosomes, and so there's a very potent small molecule ATPase inhibitor that inhibits BAF. And when you do that, you very quickly lose chromatin accessibility at thousands of regions across the genome. And so this is what it looks like in our hands here. This is some data from a grad student in my lab treating the cell line GM12878, which is an ENCODE tier 1 cell line. The reason we use it is there's all kinds of genome annotation and ChIP-seq and Hi-C and other data available for this line that we can use to make sense of the genome regions that do and don't lose chromatin accessibility. So what we've done here in these heat maps, each row is one accessible chromatin region mapped by a TAC-seq in the untreated cells. And the rows are consistent across these panels, each row being one region of accessible chromatin. And we've got them categorized here into promoters based on this histone modification signature of H3K4 trimethylation and H3K27 acetylation, either promoters or enhancers. And we have the enhancers further subcategorized into active versus poised enhancers. And again, this is done based on the histone modification. So this histone modification ChIP-seq data is from ENCODE. This is just reference data to help us separate these lines. The data on the, separate these regions, excuse me. The data on the left is our TAC-seq data. And you can see here on the left is the control. These are untreated GM12878. And when we treat with this BAF inhibitor within 24 hours, you lose accessibility pretty dramatically at many thousands of regions across the genome. But as you may see here, and of interest to us, is we don't lose accessibility everywhere, right? Promoters seem to be pretty unaffected by the loss or inhibition of BAF, whereas enhancers tend to be quite dramatically affected. We can quantify this here. You can see promoters on average really don't lose. Here I'm plotting the log2 fold change on the y-axis, whereas enhancers do lose accessibility. And in fact, they don't even lose accessibility evenly. It's the poised enhancers that seem especially sensitive to BAF inhibition. And these are enhancers that we don't think are actively enhancing transcription, but some of the machinery that's needed is already there, and they're sort of poised, we think, to respond to developmental or environmental cues. This is sort of the classic understanding of poised enhancers, which we identify based on this chromatin signature. So just some interesting insights that, thousands of regions require BAF to maintain accessible chromatin, but not all equally. One other little interesting piece of data here is that here, just showing an example region, this is a gene we know to be active in this cell line GM12878. The black tracks here are genome tracks showing you a TAC-seq data. And you can see these green regions that I've highlighted, these are promoters, we think. Probably this is the main promoter of this gene. Maybe this is an alternative promoter here. We can see that with BAF treatment, even at 24 hours, these don't change much in their accessibility. We still see these peaks of accessibility. Contrast that to these enhancers where we lose accessibility at 24 hours, but you can see the loss is extremely rapid. So within 30 minutes, we haven't looked yet earlier than that, but within 30 minutes, some of these enhancers already completely lost their chromatin accessibility or near completely, and it stays throughout the course of treatment. And in some cases, it does seem to be progressively losing accessibility, but already at 30 minutes at all these regions, we see effects. So very potent, or said another way, it really demonstrates the very acute need of these chromatin remodelers to maintain chromatin accessibility and the sensitivity of cells if you don't have that activity present. The second background point I want to give you about chromatin remodeling is that it's very dynamic during development, particularly at regulatory DNA. And as a result of that, it's also very dependent on cell types. So different cell types have very different regions of chromatin accessibility, particularly at enhancers, the sort of distal regulatory elements. And again, I just want to show some data to give you a kind of concrete example of this. So this is actually a bit of published data where we profile chromatin accessibility across a big window of mouse development from about 9.5 days, or excuse me, here, 10.5 days post-conception. This E10.5 just means the 10.5th day of gestation up to the day of birth. And at each of these stages, we profiled a different tissue or a panel of different tissues. And I'm just highlighting for you here some data taken from forebrain in this window from about 11.5 days post-conception to 16.5. This is a really critical window when neurons are differentiating. This is when you see the first maturing and mature neurons start to emerge, at least in the forebrain. And what you can see at this region here that I'm showing as an example, this is a gene called NeuroD6, encodes a transcription factor that's important for neuronal cell differentiation. And you can see that as we proceed in development, the accessibility is gained over time at the gene's promoter. And we think over here is probably an enhancer where you can see accessibility gained. And I note that this is very cell type, or in this case, tissue dependent. So if we contrast forebrain at E15.5 to these other tissues, we see that they do not have accessibility at this locus. So just to show you that this process is really highly dynamic and we think very important in development. And one of the key proofs that it's important, and the last background point that I want to give you, is that chromatin remodelers really are, we have a lot of data now to show that they are very critical for healthy development and that they are often mutated in human disease. And so a little bit of data again here to illustrate this point. So I'm showing you here numbers from a big consortium effort called the International Mouse Phenotyping Consortium. This is an effort to knock out every gene essentially in the mouse genome. They're up to now somewhere about half of the protein coding genes, about 10,000 different genes. And what I'm doing here is aggregating those numbers and just looking at the simple phenotype of embryonic lethality versus viability. And there's a phenotype you'll see here called subviable. That means embryonic lethal, but with incomplete penetrance. So you do get some embryos born, but not in the ratios that you expect. And those embryos that are born in the case of subviable are often very sick. So it's sort of a midpoint between completely embryonic lethal and viable. And what you can see is that if you take kind of any protein coding gene on average, about 30 to 35% of them either cause lethality or subviability when you knock them out. So we would call these essential genes at the developmental level. But look at the numbers if you consider the subset of these protein coding genes that are chromatin remodelers. That number jumps to almost 80%. So the vast majority of these chromatin remodelers, at least the ones have been tested, which now are up over about 40, are essential for development. In other words, if you lose both copies, you do not survive to term. And we have evidence that the same thing is true in humans. Of course, we can't make human knockouts, but we can look at data from the NOMAD consortium, for example, where they are sequencing millions now, at least hundreds of thousands of exomes and whole genomes. And they've generated these loss of function intolerance scores. So basically genes where we do not see loss of function mutations in the human population. If you sequence enough people, you can infer that that's probably because development is inconsistent with having loss of function mutations in those genes. And we see that for most protein coding genes, that they are quite tolerant, at least on average, of a single copy loss of function mutation. Contrast that to chromatin remodelers that are very, very often almost completely intolerant of loss of function mutations. For many of these genes, you essentially never see even monoallelic loss of function mutations in the sort of unaffected control population. Now, as you might expect, many of these chromatin remodelers also have kind of rare Mendelian developmental syndromes associated with them. So again, looking at percentages of genes, about 20-something% of all protein coding genes are known to have Mendelian developmental phenotypes associated with them. For chromatin remodelers, it's almost 40%. And we think this is particularly true for neurodevelopmental disorders. So many of these Mendelian disorders that are caused by mutations in chromatin remodelers have in some form of intellectual disability as a feature. But if we look at more kind of idiopathic neurodevelopmental phenotypes like autism spectrum disorder or idiopathic intellectual disability, in those patients, you also often find de novo mutations in chromatin remodeler genes much more frequently than you find de novo mutations in just your average protein coding gene. So many of these chromatin remodelers, about 15% of them are among the autism spectrum disorder Safari tier 1 risk genes. So really, I think very important group of genes. And so when I set out to start my lab in 2021, I was really interested in better understanding how mutations in these chromatin regulators, which many of them are expressed ubiquitously, how do they give rise to these different developmental phenotypes, and particularly neurodevelopmental phenotypes? And as a genomics person, I really want to understand this at the genomic level. So the sort of way I think about this is that, you know, you have these chromatin remodelers that in their normal function are remodeling chromatin at usually regulatory elements that are leading to some gene regulation and healthy development, ideally. And when you have a mutation, at least in the chromatin remodelers that are involved in this process, you get aberrant remodeling at these regulatory regions, you have gene dysregulation, and that's what causes these phenotypes. And we want to know, you know, what are the regions of the genome that rely on which chromatin remodelers for their chromatin accessibility. You can see that chromatin remodelers seem to have some specificity to which regions of the genome they remodel and in what cell types and what contexts. And when they malfunction, when you have mutations or potentially even environmental perturbations, what are the genes and pathways that are dysregulated? And again, in what cell types? And I think we're really poised as a field from a technological standpoint to answer some of these questions. So let me tell you about the experimental pipeline now that we're building to hopefully answer some of these questions using mouse as a developmental model. And this is a project in which I teamed up with some great collaborators. One collaborator at the Jackson Laboratory, Dr. Kevin Peterson, and another collaborator and former colleague of mine at UCSD, Dr. Alan Wong. And what we set out to do was recognize that there are already a number of great reagents out there. So IMPC, this mouse phenotyping consortium, has already made knockouts for dozens of chromatin remodelers. And in many cases, because they're lethal, often late in gestation or even just after birth, they're not phenotyped much after that. They say, well, this is a lethal line, and it gets put to the side, and other genes with, you know, adult phenotypes get prioritized for the phenotyping pipelines. And so we said, why don't we take those lines that are already there and look in development? We can get embryonic tissue prior to lethality and actually ask what is going on to the epigenomes and the transcriptomes and just the cells in these embryos. We are enrolling on the order of 10 to 15 genes in this study. So far, we have about 12 enrolled, and we prioritize chromatin remodelers that have a severe mouse phenotype. Often that's lethality or subviability, but sometimes it's viability with very striking phenotypes, and also lines that have human Mendelian disease associated with them. And all the lines we've gotten so far fit both of these. And our pipeline looks like this. So we harvest embryonic tissue at E9.5, E12.5, and E15.5. And again, this kind of brackets a really key kind of window in basic neuronal cell differentiation. There's a lot that goes on after E15.5 too, but this is when you start to see the first differentiated postmitotic neurons occurring in the mouse brain. And our pipeline is set up such that we do F1 crosses, and in those litters, we have litter mates that are wild type, heterozygous for the knockout or homozygous for the knockout. So we can get these really nicely controlled sets of embryos to compare to each other that were in utero together. At E9.5, we do this for whole embryos. At E12 and E15.5, we micro dissect panels of tissues, including forebrain and others. And what we do is we freeze all of these either whole embryos or embryonic tissue down and send off a little bit of material to get genotyped and to determine the sex. And then we can go back and pick which embryos and tissues we want to push forward for full molecular phenotyping, which in our case includes 10x multi-omic phenotyping, where we get single nuclear RNA-seq and single nuclear attack-seq to map open chromatin in individual nuclei. And I should mention one of the things that we're doing here is these assays are still very expensive. We can't afford to profile every tissue and every embryo we harvest here. So we're working on using bulk RNA-seq as a kind of screening tool to prioritize lines and tissues. And in the last part, I'll show you what some of that looks like. But for this bulk of this part of the talk, I want to show you just some preliminary data we have from both E9.5 whole embryos and from E15.5 forebrains. This is single cell multi-omic data, most of it coming from wild type still. And it differs from some published data in a couple important ways. So there are now a lot of really great developmental kind of reference single cell data sets that are out there. What's a little different about this is, one is this is joint from the same nucleus RNA and attack-seq, whereas most of the data that's out there is just RNA, or in some cases just attack-seq. The other thing that's different here is we're analyzing and assaying individual single embryos. And this took us a little time to work out because 9.5 mouse embryos about the size of a grain of rice, maybe a little smaller. And we want to be able to genotype, sex that that embryo and assay that individual embryo, multiple embryos individually, and then analyze in a joint space. And so let me show you some data for E9.5. Here's what it looks like when I do that. And I know this is a sort of busy slide, so let me walk you through it a little bit. Hopefully you've seen before these UMAP dimensionality reduction plots, which is a very common way to represent single cell omics data. In this case, this is that joint multi-ohm RNA and attack data. And just to kind of refresh your view here, each dot in this plot is a single nucleus, and they're roughly organized in this very, very highly reduced dimensional space, roughly organized by the similarities in their transcriptomes and accessible chromatin landscape. So that cells that are closer together here roughly are more similar than those that are further apart. And you can see a couple of things here that I want to point out. One is that we've, in a higher dimensional space, in about 50-dimensional PCA space, we've called clusters here, which is kind of part of standard single cell workflow. We've identified 45 clusters of cells that are similar to each other. And I've colored those clusters here based on these kind of broad lineage categories. So the clusters that we think are neural clusters are colored in green. The other big group of clusters here are the ones that are covered in blue. We think those are mesodermal clusters. There are some clear other epithelial cell clusters here, primitive erythroid cells, which are very distinct transcriptionally from these other cells and cluster out here. And then we have some endothelial cells here. These are the sort of broad categories. And then within each category, you can see we have some definitions of these individual cell clusters based on marker gene expression. And I'll show you some examples of that in a minute. One point I want to make about this is that, as I mentioned, these are individual embryos that are assayed separately, but they're analyzed jointly in this co-embedded space so that we can make distinctions or make comparisons between mutant and wild type. So in the case of this plot I'm showing you here, there are 20 different data sets, 20 embryos all plotted here together, a total of about 145,000 nuclei. I'm not showing you the distinctions between the embryos here, but we can individually address every embryo in this data set. And it's important to confirm, for example, that this clustering is not driven by one embryo clustering here and another one clustering here. These are clustered by shared biology, by cell type or cell state in some way. And each embryo has some representation across all of those clusters. And hopefully you can imagine that once we start mixing in mutants into this space, we can start to draw some really nice fine-grained distinctions. And I'll show you a little bit of that. So just to give you a flavor of what it looks like when we look at kind of marker genes and how we label these clusters, we don't do this based on any one marker gene. We do this based on larger sets, but I'm just showing you some examples here just for illustration purposes. So this cluster 29 down here, which are cardiac progenitors, we think this is the first and second heart field cells down here in cluster 29. And we can see that they express this canonical marker of cardiac progenitor cells, GATA4. Up here, many of these neural progenitor cells express SOX2, which is a marker of neuroprogenitors. We can contrast that here with the more committed neuroblasts. These are more maturing neurons that express TOB3. We have SOX10 expressed here in the neural crest. FLIP1 in the endothelial cells here, EPCAM expressed here in these epithelial cells. And hopefully, you can kind of get a picture from this that as we consider numbers of known marker genes, we can start to assign cluster identities to these cells. And I'll show you some other ways that we do that. Of course, we don't just have RNA data here, we have ATAC-seq data, and that allows us to do some other really cool things in leveraging this data. One thing is that we don't have to look just at individual chromatin regions. We can look at motif enrichment across accessible chromatin across entire cells. So when we do that, it looks like this. So to give you an example, this is the motif for GATA4. And if we ask for each cell within that cell's accessible chromatin regions across the genome, what is the level of enrichment for this motif in those accessible chromatin regions versus what you would expect by chance in a background set. And you can see that the GATA motif is enriched in accessible chromatin in the same cells where GATA4 is expressed, showing us, that we have the transcription factor there, and also that its binding sites, presumably at enhancers and other regions, are accessible and likely responding to it. You may notice that there are some other cell types that kind of light up with enrichment here. These are the erythroid cells that seem also to have enrichment. Those cells, the primitive erythroid cells, have a transcription factor GATA1. That's very, very important and binds to a very similar motif. So you can see how having the transcription factor expression and the motif helps you really narrow down what's going on. So you can kind of call GATA4 here and know that it might be another GATA family member out here in the primitive erythroids. In this case, we have a good sense of what that is. Just another example with SOX2, enrichment for the SOX2 motif, largely in these neural progenitor cells matching up with where SOX2 is expressed. In the more committed neuroblast, TOB3 is not a transcription factor, but a transcription factor that is expressed in those cells is called NeuroD1. We can see enrichment for its motif very specifically in those populations. So hopefully this can illustrate for you that we can get a pretty good kind of granular look at both the transcriptome and the epigenome of these cells. Here, as I mentioned, we're not looking at individual accessible chromatin regions, but we can do that. So here is a couple of just example regions. These regions I'm showing you here on the left, these are two different regions of the genome. These are known mouse developmental forebrain enhancers. These images on the bottom come from the VISTA database, which is a database of validated enhancer sequences. The blue here is showing reporter gene, LAC-Z reporter gene activity that's driven by a test enhancer, in this case, this region, and in this case, this region. So these are known forebrain enhancers. And when we look in our data, for example, in cluster 1, if we aggregate the ATAC-seq data for all of cluster 1 into this signal that we call a sort of pseudo bulk signal, you can see there's a lot of accessibility right at this region. That's a known enhancer, and at this one as well. That's also true in these more committed neuroblasts, but not as accessible in these non-neuronal cell types here. And you can contrast that here with known heart enhancers. I'm highlighting two of them here that are accessible in the cardiomyocyte progenitors, but not so much in the other cell types. So again, just two different ways that we can use this chromatin accessibility data to look at regulatory pathways and kind of global motif enrichments, and to look at individual enhancers or other accessible regions that may be affected. One other thing I want to show you in the 9.5 data before I shift gears a little bit is that one of the difficult things in single cell data is that we sort of lose the spatial context of where these cells are, right? To generate single cell data, we're disaggregating, in this case, a whole embryo, assaying individual cells. We assay about 10,000 cells per embryo. So it's a sample of cells from that embryo. And you're losing the spatial context of where in the embryo they come from. But there's now some great data and tools with which we can recapture some of that by inference. So I'm going to show you an example of that here using a spatial genomic data set, a spatial transcriptomic data set. This is a technique called stereoseq, where you actually place a section of a mouse embryo down on a slide, and you can capture the mRNAs that are expressed at different points on that slide. This is published data that we're making use from here, and I'm just showing you this is that E9.5 embryo slice from the published data, and at each point on this slide, you can see the number of transcripts that were detected, sometimes in the many thousands and tens of thousands. And so we can look, for example, if we look at a cardiac progenitor cell marker like GATA4, no surprise that in the spatial data, it's expressed in the heart. And we can see it even better if we look at some of these other markers like myosin heavy chain 6 and titin, very clear expression in the heart area of these E9.5 embryos. And, we can see that in our single cell data in this cardiomyocyte progenitor population here, cardiac progenitor population, we have expression of these same genes. And we can systematically now kind of infer the spatial positioning of clusters like cluster 29 using a nice tool from Aviv Regev's lab called Tangram that uses deep learning to make the assumption that both a spatial data set and single cell data set have some shared underlying biology and gene regulation space. And so you can try to infer, if you have a spatial data set, where in that spatial data set your single cells would have most likely originated from. And you end up with a sort of spatial probability score for this cluster of cells. You can see that when we do this for cluster 29, the cardiac progenitor cells, these map really, really nicely onto the heart. And this works well, not just for the heart. I'll show you some brain clusters here. So this cluster 1 are what we think are telencephalic neuronal progenitors, a very early stage, probably radial glia or neuroepithelial cells. I'm just insetting here where those cells are in the UMAP right here. The green are the cells in this cluster 1. And when we map those to the spatial transcriptomic map, you can see they map right to the forebrain. And this aligns really nicely with the expression patterns of known marker genes like FOXG1, for example, that are very specifically expressed in that cluster in the UMAP space. And this is an in situ for FOXG1, which lines up really nicely with the spatial inference. And here are just some more examples. We can do this with midbrain cluster here. We think these are midbrain neural progenitors. Here they are in the UMAP space marked by EN2, and here's EN2's expression from RNA in situ. Similar here for floor plate organizers that are here in the UMAP. They express sonic hedgehog, which is known to be expressed along the floor plate. So all of this to say that I think, with this single set of experiments, we get a really deep look into what's happening at the transcriptomic, epigenomic level in ways that we can infer spatially back to the embryo. And we're really excited about the granularity of what we're going to be able to learn as we start to process more and more mutants through this pipeline, especially now that we have a really robust kind of reference map built up from our data. In the last bit here, I just want to share with you a little bit of E15.5 data. I'm not going to go in as much depth, but I'm going to touch on that in the last part of the talk. So I just want to introduce a little bit of it here. So the difference here is now we're looking just at forebrain. This is not whole embryo. This is just E15.5 forebrain. And in this UMAP plot, kind of same layout, 10 embryos shown here, about 75,000 nuclei. But the coloring here is different. These are all or mostly neuronal cell types. We're looking in the brain later in development. The main bifurcation here is glutamatergic neurons and their progenitors in green versus GABAergic neurons and their progenitors in blue. We do have some other cell types that have made their way in here, some vascular, some immune, et cetera, but most of these are neurons or neuronal progenitors. And I'm not going to focus too much on what's going on here. This is relatively new data, but I do want to show you one thing that really stands out, and that is these set of clusters here. which we think really nicely captures cortical differentiation of glutamatergic neurons. So let me show you a little bit more about what I mean by that. So the cortex is this outer layer of the mouse forebrain. Cortex in mouse and humans, very important for higher level cognitive functioning. And it's very well studied as a very characteristic layer by layer organization that extends from the inner portion here where there's a ventricle to the outer portion here where there's the pia. And the interesting thing, or one of many interesting things about the cortex at this stage, is that differentiation of neurons is stratified spatially in the cortex in the following way. At the ventricular zone here, all the way down by the ventricle in these base layers, this is where you have the neuronal progenitors, what are called radial glia that express SOX2. These are the progenitors not only of neurons, but also of oligodendrocytes and astrocytes later in development. At this stage, they're really still mostly generating neurons. And as that differentiation proceeds, the first step is to proceed to a state called an intermediate progenitor. These are marked by a gene called eomes and others. And those are in this subventricular zone. So they're not quite as far down into the ventricular zone. And then from there, as the neurons differentiate and become postmitotic, they migrate up towards the pia in layers as they become more differentiated. And they populate first in the outer layers and then in the middle layers and then towards these deeper layers. And we can see this really nicely in our data. Let me show you. So I mentioned that these radial glia in the ventricular zone are marked by SOX2. So I'm just isolating in the UMAP space here, these cells in green, going from radial glia here to the more differentiated neurons on the other side here in layers. And you can see that SOX2 is expressed up in this end, in the radial glia. And if we look at mRNA and C2 hybridization, this comes from the Allen Brain Atlas data. You can see SOX2 expressed all the way down here in the ventricular zone. And if we look at the spatial projection of this cluster, cluster 1, onto E15.5 mouse forebrain spatial data, we can see that the cluster projects to the ventricular area, to the base here. As we move up the zone, we'll see eomes marked intermediate progenitors here in the middle. These are these cluster 2. Here's an mRNA and C2 of eomes a little bit higher in this ventricle to pia kind of axis here. Here's where cluster 2 spatially maps, again, a little bit higher. And now we start to move into some of these more mature neurons and maturing neurons, SATB2 being one marker of those. And SATB2, you can see, really lights up this cluster 3 here. You can see it in the mid. This is really, I think, one of the deeper layers of differentiating neurons that we have here. You can see where it maps cluster 3 right to the middle of this kind of cortical space here. And I'll show you an example of an upper marker, TLE4. This one happens to be in cluster 15, the most upper here. It's towards the outer portion of where these neurons are migrating, more towards the pia than the ventricle. And you see that this cluster 15 spatially maps here to the outer portion. So I think you can hopefully see the kind of granularity with which in this one assay we can really pick apart, and this is just kind of a first pass at this data without even doing things like pseudo time analysis yet. This is just off the base clustering. We can really see these developmental processes happening. And I highlighted this one, number one, because it's well characterized, but also because I'm going to come back to it in the last part of the talk, where I tell you about some of our early insights on members of the BAF family of chromatin remodelers. So just to remind you what I set out at the start, which is that we're interested in this chromatin remodeling family called BAF, that if you inhibit it, you lose chromatin accessibility at thousands of regions in the genome. And I'm going to show you here a diagram of BAF that includes a little more information on all of the subunits. This is the, quote, canonical version of the BAF complex. And you don't have to know all of these subunits for our purpose here. But the reason I bring this up is that, whoops, let's see, something isn't showing up here. Let me see if I can get this. There we go. So the reason I bring this up is that a number of these subunits, if they are mutated, any one of these individual subunits, they can cause a rare Mendelian pleiotropic disorder. It's actually a group of disorders that are referred to as switch-SNF-related intellectual disability disorders. It's an awful acronym, SSRIDDs. It's descriptive, but just a mouthful. Sometimes this is also known as Coffin-Siris syndrome, but these patients have very severe intellectual disability. They have some distinctive facial features, abnormal hair growth, other features. And these seem to be caused, they're almost always de novo monoallelic mutations. So they're autosomal dominant, but very rarely actually passed on. And interestingly, several of these genes also turn up in patients who have non-syndromic autism, or at least haven't been diagnosed with any syndrome, but have been diagnosed with non-syndromic autism and intellectual disability. The ones that I've starred here or put an asterisk, these are tier 1 Safari autism risk genes. And in fact, one of these genes alone, ARID1B, this one here, is thought to account for about 1% of all intellectual disability cases. So these are rare, but they're not that rare. And this has been of interest to us, to my lab, to others, but it became of extra interest when in 2018, a new version of BAF was discovered, which is sometimes called the non-canonical BAF or NC-BAF complex. And it shares many of the same subunits with the quote canonical BAF, except for a couple. And the one I want to highlight today is this one. Sorry about how these check marks are all showing up. The one I want to highlight today is this one called BICRA. There are two proteins, paralogs, that can occupy this place in the complex, one called BICRA, one called BICRA-L. I'm going to focus on BICRA today. They're coded by two separate genes. They're not isoforms, they're paralogs. So this BICRA gene, shortly after this complex was discovered, it was found that patients, sorry, I got some animation issues here. There we go. That patients diagnosed with these SSRIDDs who didn't show mutations in any of these canonical BAF subunits, sometimes, we now know of about a dozen cases like this that are documented in the literature, have mutations in BICRA, which was really interesting because it implicates for the first time this non-canonical BAF complex in disease for sure, but in brain development at all. And it's still a big mystery what this non-canonical BAF is doing, what BICRA is doing. We have very little understanding. So we figured this was a good place for us to jump right in. And as I mentioned, we're starting to use bulk RNA-seq as a kind of screening tool before doing single cell on everything. So I'm going to show you here the results of one of those experiments where we've taken 4 brain from E15.5 mice, wild type, and in this case, 5 different mutant lines. These include several subunits of knockout lines for subunits of canonical BAF that are implicated in neurodevelopmental disorders, and BICRA, and one other gene that is not a BAF subunit. And what you can see, even at a high level, just from the PCA space, is that these BICRA samples cluster right out in the PCA space, suggesting that there's something about the transcriptomes of these 4 brains that really distinguishes them, not just from wild type, but also from these other mutants. And so we can perform kind of a standard differential gene expression analysis to look at what goes up and down in these BICRA mutants versus wild type. We're not super well powered here. We have 3 knockout 4 brains and a bunch of wild types, and we have sex as a variable here that we're controlling for. But we get, hundreds of genes that are up and down. And here's where I think the most interesting thing to me jumps out, which is that if we look back at our kind of cortical glutamatergic differentiation trajectory here, where, to remind you, we're going from this cluster here, which are the radial glia, those base level progenitors, to the more upper level differentiated neurons through these intermediate progenitors. We can do a gene set enrichment analysis, and in this case, we're using a package, really nice package called UCell, to take the set of genes that are down-regulated in the BICRA mutant. So these are genes whose expression goes down when you lose BICRA, and ask where are those genes expressed, and they're expressed on average, they're enriched anyway, in these differentiated upper level glutamatergic neurons. Contrast that to the genes that show increased expression in the BICRA mutants. Those map to the radial glia progenitors and the intermediate progenitors. So we don't know whether these are gene sets that are changing within cells, or what we have here is a delay of cortical differentiation or some kind of defect such that you have more progenitors and fewer differentiated neurons in the mutant. I'd say that's probably the most likely thing, but this is the kind of screening that now we have fast-track BICRA for single-cell phenotyping, which unfortunately the data is not back yet, but those assays are being performed like literally right now this week. So hopefully it gives you a flavor of where we're hoping to go and how we're working this bulk RNA-seq into this workflow to try and prioritize lines and tissues. So conclusions for now are, I think we have a really nice pipeline in place to phenotype the effects of chromatin remodeler loss in multiple stages of development for our purposes, particularly focused on neuronal development because of the role of these genes in neurodevelopmental disease. We've enrolled 12 mutant lines to date. They're at various stages of breeding and sample collection and assay, et cetera. And some preliminary data we have, aside from the kind of reference atlases that we've generated of cell types, are that we think in these BICRA mutants, which are the first time people have reported a neuronal phenotype in these mutants, we can see a defect in cortical differentiation, which implicates not only BICRA, but the non-canonical BAF complex in brain development, we think, in a new way that we're digging into on multiple fronts, including iPSC lines and mice. And so with that, I'll just acknowledge some great collaborators at JAX, at UCSD, and some folks in my lab, highlighting here 3 PhD students, wonderful PhD students, some of whose work I spoke about today. And if there's time, I'll be happy to take questions and have some discussion.
Thank you so much. Yes, we'll kind of open it up for discussions. If you'd like, you're welcome to unmute and ask a question or type a question into the chat.
So hi, Dr. Gorkin. I'm Cody Dunton. I'm a postdoc in Vadim Bachman's lab. The talk, I think, is phenomenal. The technologies, I think, show a lot of potential and powerful data that could be generated. I think one thing of specific interest for me was the spatial, the ability to look at this spatial resolution, more or less, of some of these techniques. But one thing that I wanted to ask is about the incorporation of other next generation sequencing techniques as well. So say ChIP-seq data. And then one thing that we work quite a bit with is Hi-C. I know it's a different type of output, but it still maps to the genome. Do you think that those are things that could be incorporated into these readouts as well?
Yeah, we are really interested in all of those modalities. I think, you know, we have focused on single cell chromatin accessibility or single nuclear chromatin accessibility for a couple of reasons. One, it's the most kind of mature, I think, of the single cell epigenomic suite. And the other is that it actually is the molecular phenotype of the genes that we're studying. You know, it's their direct product, I guess you could say, on the epigenome. We are looking into ways to incorporate either ChIP-seq or probably more likely cut and tag or cut and run into this. We've done both in cell lines and in bulk tissue, but not at the single cell level yet. So in that developmental trajectory I showed you where we have E10 to E to P0, we have ChIP-seq data for histone modifications in that data series. We also have high ChIP data. We don't have high C, but we have some high ChIP data that is not published yet, but going to be published soon by my postdoc lab, that has at least some of those tissues and time points covered. So the short of it is I'm very interested in integrating that. I can't say I have a super clear vision yet of how to infer cell type specificity from bulk, you know, ChIP-seq or bulk Hi-C. I'm excited about things like the single cell methyl Hi-C methods where you can cluster cells based on methylation patterns and then look at the Hi-C patterns, because I don't think clustering cells based on Hi-C alone is going to give you great cell type resolution. So some different kinds of answers there, but hopefully that fleshed out my thoughts on integrating these other data sets. But I think it's totally something we're interested in.
Yeah, definitely. And thank you for the response. And I agree. I don't think that you can map cells based on Hi-C data, but looking at the Hi-C maps from cluster data, I think has a lot of power. And obviously, being from Vadim's lab, we are very interested in chromatin packing. And so that's mainly the reason that we want to incorporate things like ChIP-seq data and Hi-C mapping into some of these outputs. But yeah, I think that this is, I have not seen this before. This is really, really cool stuff. Thank you.
Thanks, Cody. And one other thing of potential interest I'll mention, because I also am really into 3D genome organization and have worked on that in my postdoc some, but an angle that it connects here is that this non-canonical BAF complex that I mentioned if you do a cut and run for the subunits that are specific to that complex as opposed to canonical BAF, you find it enriched at CTCF binding sites. So we think it may have something to do with 3D genome organization. We don't know that for sure, but we have experiments going on in the lab, for example, to drug that complex in particular. We have drugs against BRD9 and do high C to look for, and CTCF chip and other things to look for potential effects on 3D genome. I think those experiments are proving kind of a little bit tricky to tease out the effects of the different BAF complexes because there's some functional crosstalk between them. They can compensate for each other. When you lose the function of non-canonical, canonical can compensate, and vice versa. So we don't have answers there yet, but there's a potential through line there to 3D genome organization that you might be interested in.
Yeah, that's really, really cool. And just to touch on briefly, so you mentioned that canonical can compensate for non-canonical. So in some of these knockouts, where you do say delete a gene that's important in non-canonical, that still leads to the outcomes that you see. So I guess in that case, non-canonical BAF is not compensating for the genes that are being knocked out in that case.
Right, it's not a full compensation. So a lot of these subunits are very dosage sensitive. So, you know, in humans, if you lose, these are mostly loss of function mutations. If you lose a single copy of a single subunit, you can have very, very severe intellectual disability. And for some of those subunits, if you lose a single copy of a single subunit, you're not born. The functional redundancy that I'm referencing really comes more from the study of cancer. So these chromatin remodelers are very frequently mutated in cancer. My lab isn't so focused there, but other groups are doing great work there. And what they've shown is that in cancers that have canonical BAF mutations, that are, the canonical BAF is thought to be a tumor suppressor. So you get biallelic mutations in at least some subunits of the complex. And the non-canonical BAF are synthetic lethal dependencies in those cancers. So if you have a cancer that's canonical BAF mutant and you can drug non-canonical BAF subunits, you can kill those cancer cells because there's some amount of compensation going on. But clearly from the developmental data, it's not a full functional compensation or redundancy, but it's partial.
Okay, thank you. And thank you for the presentation. Again, this data is really, really exciting.
Thank you. Thanks for the question.